Today we are going to learn about the prokaryotic cell and eukaryotic cell. Although it's a very simple topic and you all are very much aware about the differences between the prokaryotic and the eukaryotic cell, but we'll try to see some of the basic differences, and we'll try to see that what sort of questions will be asked from this particular section or this particular topic. So first is the difference between the prokaryotic cell and eukaryotic cell. So you are very well aware that this term prokaryotic and eukaryotic arise due to the one particular organelle, that is nucleus, right? So in the case of prokaryotic cell, nucleus is absent, while in the case of eukaryotic cell, nucleus is present. And you know what is the function of nucleus? In the eukaryotic cell, nucleus is a double membranous organelle in the case of eukaryotic cell, which contain the genetic material. So genetic material lies within the nucleus in the case of eukaryotic cell. This particular genetic material is the main genetic material of the cell, while in two other organelles, if you say in the case of plant, chloroplast as well as mitochondria, they also have their genetic material within their compartment. Chloroplast has its own DNA and the mitochondria has its own DNA. But still, mitochondria and chloroplast, both of these organelles depend upon the nuclear DNA also for protein synthesis, for the synthesis of certain enzymes. Right? So in the case of eukaryotic cell, we have a well-defined nucleus, while in the case of prokaryotic cell, well-defined nucleus is absent. Here, genetic material lies freely in the cell cytoplasm. So this is the basis of defining prokaryotic and eukaryotic cell. The word eu means true and karyo means nucleus. So in the case of eukaryotic cell, we have a true nucleus, while in the case of prokaryotic cell, nucleus is absent, right? So we'll see some of the differences. So the prokaryotic cell, they have a simple organization, while eukaryotic cell have a complex organization. Simple organization means here, if you will see the point number 4, membrane bound organelles are absent in the case of prokaryotic cell, while they are present in the case of eukaryotic cell. So many structures are there in the case of eukaryotic cell which make it complex. Well, in the case of prokaryotic cell, you will not find such membrane bound organelle. Right? So membrane bound organelle are absent in the case of prokaryotic cell, which make it simple in organization. While in the case of eukaryotic cell, we have the single membrane bound organelles as well as double membrane bound organelle. Okay. So size of the cell, prokaryotic cell is typically small, 0.2.
Excuse me, sir.
Yeah, tell me.
Sir, can you please change the slide, sir?
Yeah. Hello.
हाँ sir.
हाँ, बताइए.
Sir, now it is in first slide, sir. Can you change the slide, sir?
I have
Second.
changed. Now it has come.
No, sir. We are in first slide.
Okay, you are in first slide only. It's not changing.
Yes.
Okay, just a minute.
No, sir.
Then I will. Okay, 1 minute. Now it is visible. Slide number 2.
Sir. Now it's visible, sir. Continue,
okay.
sir.
Okay, fine. Thank you so much. Thank you.
Sir.
So, prokaryotic cell has a simple organization, while eukaryotic cell has a complex organization. Now, size of the cell, if we will see, the prokaryotic cell are smaller in size as compared to eukaryotic cell, and it is very natural. Why it is very natural? Because in a eukaryotic cell, you have number of double membrane as well as single membrane organelle within the cell. So these organelle need space to be there, right? if these organelles are present, definitely they need space, and definitely the size of the eukaryotic cell should be larger than the prokaryotic cell. Okay, so one basic difference is nucleus. In the case of prokaryotic cell, it is absent. Genetic material lies free in the cell cytoplasm, while in the case of eukaryotic cell, it is well defined and genetic material is present within the nucleus. Now we will see cell wall. Cell wall in the case of prokaryotic cell, usually it is present, but still there are certain prokaryotic cell which lack a cell wall. And if it is there, it is chemically complex and usually made up of peptidoglycan. We will see what is a peptidoglycan made up of in our later slide. Then in the case of eukaryotic cell, in some cell wall is present, while in some it is absent. For example, in the case of plant, it is there, it is made up of cellulose. In the case of fungi, it is made up of chitin, while in the case of animal cell, cell wall is absent. Then membrane bound organelle are absent in the case of prokaryotic cell, but they are there in the eukaryotic cell. Then we will see the ribosome, the protein machinery or the protein body, which will synthesize protein. Then in the case of prokaryotic cell, we have 70S ribosome, smaller ribosome. Where in the case of eukaryotic cell, we have both smaller as well as larger ribosome. So 70S ribosome is present in the chloroplast as well as mitochondria, that is the cell organelle, while 80S ribosome is present in the cell cytoplasm. If you will see the process of cell division, so in the case of prokaryotic cell, mitosis is not there. Like we have in the case of eukaryotic system, a well-defined mechanism by which parent cell divide to form two daughter cell. It is well defined. There are different phases, right? Like prophase is there, metaphase is there, anaphase is there, telophase is there. So these four phases are there in the mitosis, which allow the chromosome as well as the cell cytoplasm to divide into two daughter cells. While in the case of prokaryotic cell, such mechanism is absent. They divide through binary fission. That means when the cell size increases, then the constriction forms from the plasma membrane, from the periphery. एक अंदर की तरफ एक U शेप कंस्ट्रिक्शन फॉर्म होता है, धीरे 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 दोनों U शेप कंस्ट्रिक्शन जो है आपस में मिल जाते हैं और आपको दो बैक्टीरियल सेल्स सेपरेट हो जाते हैं फ्रॉम द पेरेंट बैक्टीरियल सेल, बट वहां पे माइटोटिक डिवीजन नहीं होता. Okay? True sexual reproduction is also absent in the case of prokaryotic cell. They have only exchange of DNA fragment. Exchange of DNA fragment occur either through the process of conjugation or through transformation or transduction, which is mediated by viruses. But like in the case of eukaryotic system, we have a defined mechanism which forms gamete, that is meiotic division. Then we have fertilization to form a zygote. Such processes are absent in the case of prokaryotic cell. Just movement of DNA fragment. Few or certain region of the DNA is moved from one bacterial cell to another bacterial cell. Like in the process of conjugation, you remember, if you recall your microbiology, then we have F plasmid, which moves from the donor bacterial cell to the recipient bacterial cell. But there is no as such movement of the chromosomal DNA. Chromosomal DNA का movement कब possible होता है? Conjugation के टाइम पर, जब आपका F plasmid जो है वो main bacterial chromosomal DNA से जाकर fuse कर जाए. And when it starts moving, then it may carry some of the chromosomal segments together. But it is never like that कि entire DNA from one bacterial cell move into another. ऐसा कभी भी नहीं होता. Just exchange of few segment or few stretch of a DNA occur from one bacterial cell to another bacterial cell. Okay. Okay. Then plasma membrane. If you will see plasma membrane in the case of prokaryotic cell, no carbohydrate. Prokaryotic cell lack carbohydrate in the plasma membrane. In their plasma membrane, carbohydrate is not there, and they also lack sterols in most of the cases, but we have exceptions. In the case of eukaryotic cell, they have sterols as well as carbohydrate, which act as a receptor. Then cytoplasmic streaming or the movement of cytoplasm is absent in the case of prokaryotic cell, while it is there in the case of eukaryotic cell. Since in the cytoplasmic streaming means it is a movement of cytoplasm. Why it is required in the case of eukaryotic cell? Because in the eukaryotic cell, we have the membrane bound organelle. And if we want to send anything from one organelle to another organelle, the cytoplasm should move. Or let us suppose during the process of photosynthesis, if chloroplast has to get localized to a place where the light will come, then they have to move towards that region where they can have light for the process of photosynthesis. So it is required in that case. But since in the case of prokaryotic cell, the organization is more or less simple. We have no such membrane bound organelle. So it simply moves from one place to another place through the process of diffusion. But we do not have cytoplasmic streaming. Eukaryotic cell, just visualize a larger compartment, a larger room where if you want to send or communicate to another corner of the room, you must have something by which you can communicate. So for the purpose of communication, either chemical communication or physical communication, we need cytoplasmic movement, which is there in the case of eukaryotic cell. Just compare a large room and a small room. So if you are in a larger hall, definitely in order to communicate from one corner to another corner, you need certain medium. But in the case of smaller room, even just you say normally, it is audible. So same thing in the case of prokaryotic and eukaryotic cell. So in the case of prokaryotic cell, cytoskeleton is absent. While in the case of eukaryotic cell, we have a complex cytoskeleton, and this cytoskeleton will also help in the movement of cell organelle from one place to another place. So here the cytoskeleton is mainly formed by microfilament, intermediate filament, and microtubules. Then if you see the process of transcription and translation, in the case of prokaryotic cell, both the process takes place simultaneously. That means transcription and translation are not separated either in space or with time. Well, in the case of eukaryotic cell, we have spatially and temporally separation between the transcription and translation. So Shreya, am I audible?
Ha, sir, you are audible, sir.
Chinta, then Shreya, I want to ask one question to Shreya. Is she there? Sangeeta? Just a minute. Hello. Yes, anybody who want to answer my question, please unmute yourself.
Yes, sir.
Okay, right. So can you please tell me, in the case of eukaryote, transcription takes place where?
Transcription takes place inside the nucleus.
Transcription, okay. And translation?
Translation happens in the cytoplasm.
Cytoplasm, sir.
Okay, very good. So in the case of eukaryotic system, we have transcription in the nucleus and translation in the cytoplasm. That means the space where both the process are occurring are separated, as well as the time when transcription is going on, translation is not happening, right? So they are separated both by space as well as time. But in the case of prokaryotic system, it is not there. As soon as the RNA polymerase synthesize mRNA molecule, translation will start, because there is no such compartment where DNA is localized. DNA lies freely in the cell cytoplasm. Now, chromosome, if you see the arrangement or the organization of chromosome, in the case of prokaryotic cell, we have a single circular chromosome and it lacks histone protein. While in the case of eukaryotic chromosome, the same amount of DNA is present in the form of multiple linear chromosome. Like if you see human cell, the entire DNA content is divided into 46 chromosomes. 44 chromosomes will be autosomes and 2 chromosomes will be sex chromosomes, right? So in the case of eukaryotic system, the entire DNA is divided into multiple linear chromosome, and chromosome formation require histone protein. That means DNA is associated with histone protein in the case of eukaryotic system, while in the case of prokaryotic system, histone proteins are absent, but researchers have told they have histone-like proteins. So proteins are there in the case of prokaryotic cell also, which are associated with the chromosome, but they are not histone protein, they are histone-like protein. Okay, so can anybody tell me whether histone charge क्या है histone के पास? It is positively charged or negatively charged? Histone protein.
Positively charged.
It is positively charged. Very good. So histone proteins are the basic proteins. DNA is negatively charged and histone is positively charged, right? Okay. So in DNA replication, in the case of prokaryotic cell, it is single repliconic. Well, in the case of eukaryotic cell, we have multiple replicon. Can anybody tell me what the word replicon means here? Multiple replicon and single replicon?
The place where the origin of replication takes place.
Okay, so in the eukaryotic cell, it is multi-repliconic. What does that mean?
It contains multiple originating stages.
Okay, that means we want to say in the case of eukaryotic system, in single chromosome, if you will see, we have multiple sites from where replication can start, right? You have seen that Y-shaped replication fork in most of the books. So in the case of eukaryotic system, we have many regions from where replication can start. That's why we are saying they are multiple repliconic. And why they need to be multiple repliconic? Why we can't be single repliconic? Why eukaryotic system need to be multiple repliconic?
Sir, if it is only one replication origin, then it takes some days to replicate DNA, sir.
Exactly. So in order to speed the process, we have multiple replicon in the case of eukaryotic system. Okay. Then transcription, in the case of prokaryotic cell, we have a single RNA polymerase. Well, in the case of eukaryotic system, we have 5 different RNA polymerase for the synthesis of different kinds of RNA molecule. Well, in the case of prokaryotic system, single RNA polymerase will synthesize all the different kind of RNA molecule. Then arrangement of genes, in the case of prokaryotic system, we commonly see the operon model. While in the case of eukaryotic system, operon are absent. So anybody can tell me what is operon? And what is the advantage of having an operon in the prokaryotic system? कोई भी?
Please explain, sir.
Okay, then I will explain. You know about operon a little bit. Operon, where we have a cluster of genes together. in the case of eukaryotic system, genes are not clustered together. Although we have gene families even in the case of eukaryotic system, but we don't have operon. In the case of operon, we have a cluster of genes which are regulated by a common regulatory sequence. That means all the genes have a single promoter sequence. They all will transcribe through the same promoter. So what is the advantage of this? If all those genes which are present in a cluster, if they are involved in certain metabolic pathway, then they all will transcribe together from a common promoter, and they all are regulated also at the common promoter. The same promoter से उनका ट्रांसक्रिप्शन भी होगा, सेम प्रमोटर से उनका रेगुलेशन भी होगा. तो आपको पता है कि अगर हम प्रोकैरियोटिक सिस्टम की बात करते हैं तो वहां पर जीनोम साइज इज स्मॉल, genome size is not very big, right? So if they have a smaller genome size, they have evolved in such a way that with lesser genome, how they can have the expression of genes. They don't want that every gene should have its own promoter. So instead of giving a regulatory sequence to all the genes which are present, the genes which are playing a role in a common metabolic pathway, they have clustered them together and they have given a single promoter. So in this way, the size of the gene is reduced. Operon आने से हर एक जीन को अपने ओन प्रमोटर की जरूरत नहीं है. एक तो रेगुलेशन इजी हो गया, बिकॉज अदरवाइज यू नीड टू रेगुलेट different genes separately, right? Having a common promoter, that means your regulation is also easy and the size of the genome is also managed. So that's why in the case of prokaryotic system, we have operon. Well, in the case of eukaryotic system, since the genome is very big, as such, we do not need operon model. Okay. So now, what are the ways to distinguish bacterial cells? So Gram staining, which was discovered by Christian Gram, so it's a way to distinguish the bacterial cell. So after Gram staining, we have typically two types of bacterial cells. Gram positive, which retain the crystal violet color of the stain, while Gram negative, which fails to retain the crystal violet color of a stain. Instead, they take the color of counter stain, and the counter stain is saffronin, which is of red color. So what is the basis of this Gram staining? That is difference in the structure of the cell wall. In the case of prokaryotic system, we have the Gram negative bacteria and the Gram positive bacteria, right? So Gram negative bacteria, which do not retain the Gram stain, while Gram positive bacteria which retain the Gram stain, and the basis is the difference in their cell wall. So in the case of Gram positive bacteria, they lack outer membrane and cell wall is multi-layered as well as thick. We have discussed in the differences while we are going through the differences. Then in the case of prokaryotic system, cell wall is complex and it is made up of peptidoglycan. So in the case of Gram positive bacteria, the cell wall is multi-layered and thick and they lack outer membrane. Well, in the case of Gram negative bacteria, outer membrane is present and it is made up of lipopolysaccharide, lipoprotein and phospholipids. And here cell wall is single or few layered. So we'll try to understand this through the help of a diagram. But before going through the diagram, let us see the structure of peptidoglycan. So in the case of Gram positive bacteria, cell wall is multi-layered and thick. That means the peptidoglycan cell wall is thick. While in the case of Gram negative bacteria, the same cell wall is thin, consists of either single layer or few layer. So this is the structure of peptidoglycan. Peptidoglycan is made up of two monomeric units which are joined together through the glycosidic bond. So one monomer is N-acetyl glucosamine and another monomer is N-acetyl muramic acid, right? So they form the glycan chain, and two glycan chain are joined to each other through the peptide bridge. And this peptide bridge is between the two NAM, N-acetylmuramic acid. And this N-acetylmuramic acid also contain a tetrapeptide chain. So these two tetrapeptide chain of N-acetylmuramic acid is joined through a peptide bridge. So likewise, the peptidoglycan constitution is there. So now we will see the difference between the gram-positive and gram-negative bacteria. So in the case of gram-positive bacteria, as you can see here, they have the cell membrane and then they have a peptidoglycan, right? And this peptidoglycan is consisting of NAM and NAG. Apart from these two monomeric units, which are joined together through the glycosidic bond, we have the teichoic acid. Teichoic acid is an important constituent of gram-positive bacteria, right? So this peptidoglycan layer in the case of gram-positive bacteria is multi-layered. It has 1, 2, 3, and 4. 4 such glycan chain are there. Okay? But in the case of gram-negative bacteria, the same peptidoglycan is very thin, right? And it has an outer membrane, and this outer membrane is consists of lipid, lipoprotein, as well as lipopolysaccharide. So when we try to stain both the gram-positive bacteria or the gram-negative bacteria by the help of the gram stain or the crystal violet, तो इसमें हम पहले क्या करते हैं, crystal violet से we treat the bacterial cell. After treating them with the crystal violet, then we will give a mordant, that is iodine. So what happened? This iodine form a complex with the crystal violet stain, and this complex is a big complex. It will not able to move outside. And another thing, after that we will treat with alcohol. So this alcohol will, what it will do, it will dehydrate the peptidoglycan. So again, the size of the pores is reduced in the case of peptidoglycan layer. So the crystal violet, which is complex with iodine, which is present within the cell, bacterial cell, does not able to move out in the case of gram-positive bacteria. Well, in the case of gram-negative bacteria, due to the presence of the outer membrane, the movement of crystal violet stain is not so easy. It will not able to reach that easily within the bacterial cell due to the presence of this barrier. Due to the presence of this barrier, आपका जो क्रिस्टल वायलेट स्टेन है वो बहुत अच्छे से, बहुत इजीली सेल साइटोप्लाज्म ऑफ द बैक्टीरिया में पहुंच नहीं पाता. फिर भी कुछ पहुंचता है. आयोडीन से जब आप फर्दर ट्रीट करते हो, ये आयोडीन उसके साथ कॉम्प्लेक्स बना लेता है और वो कॉम्प्लेक्स इज अ बिगर इन साइज. बट व्हेन वी ट्रीट इट विद अल्कोहल, तो जहां पर इन द केस ऑफ ग्राम पॉजिटिव बैक्टीरिया अल्कोहल इज डिहाइड्रेटिंग दैट पर्टिकुलर सेल वॉल, इन द नेगेटिव अल्कोहल का इफेक्ट क्या होता है? इट विल फॉर्म पोर्स, एंड ड्यू टू द फॉर्मेशन ऑफ पोर्स द iodine and the crystal violet complex moves outside the gram-negative bacteria. That's why gram-negative bacteria does not retain the crystal violet color of the gram stain. So जब आप उसको काउंटर स्टेन से स्टेन करने की कोशिश करते हो, तब आपको रेड कलर दिखाई देता है क्योंकि उसके पास क्रिस्टल वायलेट कलर स्टेन नहीं हुआ. ठीक है? So this is the basic difference between the gram-positive bacteria and gram-negative bacteria is with respect to cell wall. That means the chemical composition, presence of different types of protein, present of different types of component की वजह से they respond differently to the gram stain. Okay. So there are certain cells which we call as gram variable. Typical bacterial cell wall. So gram variable, as the name suggests, that certain gram positive bacterial cell show varied response to gram stain depending upon their age in the culture. ठीक है? मतलब ग्राम स्टेन का जो रिस्पॉन्स है वो उनकी एज के हिसाब से वेरी कर रहा है. fresh culture show positive response to gram staining, while aged culture show negative response. So such gram-positive bacterial species are known as gram variable. And the most notable example are from the Bacillus, Clostridium, and Mycobacterium species. Well, among prokaryote, there are certain bacterial species which lack cell wall or have very little wall material. So, इसमें आपके पास example है Mycoplasma, Spiroplasma, and A. coli plasma. So, due to the absence of cell wall, they can pass easily through most of the bacterial filter, and sometimes they are even confused with the viruses. So this is atypical bacterial cell wall. That means cell wall is not there. Okay. Then we are going. Okay, we have to learn something about mycoplasma. So mycoplasma are also prokaryotic. They are the bacterial cell, and they belong to the molecule class of bacteria. So molecule class of bacteria, the word molecule is derived from two Latin words. Mollis means soft or pliable, and cutis means skin. That means mycoplasma are such bacteria which lack cell wall. As we have read in the previous slide about mycoplasma, spiroplasma, and A. coli plasma. All the three classes, all the three genus, they belong to the class molecule. So they include bacteria that lack a cell wall around their cell membrane. These are the smallest, simplest, self-replicating forms of life, right? So these are smallest, even they are smaller than the normal bacteria. They are also bacteria from the bacteria which have the cell wall. They are simple and self-replicating forms of life. They contain minimum set of organelles essential for growth and replication. So they are self-replicating. They are not like viruses which require the host machinery to replicate its own DNA, while mycoplasma, although their size somehow resemble to the size of the viruses, or you can say the very large virus particles, they have a size similar to the size of the mycoplasma, but they differ from viruses in the way that they are self-replicating. They can replicate on their own. They may be a parasite, they may depend upon the host cell for the nutrient, but they do not require their machinery for the purpose of replication, like viruses, unlike viruses. Okay. So they contain plasmamembrane, they have ribosome, they have a genome consisting of a double-stranded circular DNA. They can be parasitic or they can be saprotrophic. Either they require a living cell for their survival, or they may feed on the dead cell. And they can be parasite on human, animals, and plant. Their size, as we have discussed earlier, they are very small in size. 0.3 to 0.8 micrometer in size, less than 1 micrometer. Their genome size is also small, just 580 kb to 2200 kb, which limit their metabolic capacity. Then mycoplasma is an example of regressive evolution and genome reduction. It means in the case of bacteria, we have a bit larger genome size, while these are also bacteria, but they have reduced their genome because they have dependent upon the host cell or host machinery for some of the nutrient. Instead of synthesizing on their own, they have reduced their genome and they have dependent upon the host for the recovery or for the deficiencies they have. So they are of gram-positive origin as indicated by their 16S ribosomal RNA gene sequencing. So this 16S ribosomal RNA gene sequencing is a common method to differentiate one bacterial species from another bacterial species. And this is known as DNA barcode in the case of prokaryotic system. Can anybody tell me what is the gene sequence that we use in the case of eukaryotic system for the same purpose, for DNA barcoding? Anybody know about DNA barcode? किसी को अच्छे से समझ में आया? What do we mean by DNA barcode? Anybody of you? चिंता?
No, sir.
अच्छा, डीएनए बारकोड का मतलब क्या है, पहले हमको ये समझना पड़ेगा. We know that all living form have genetic material and most of us have DNA as a genetic material, but there are exceptions. Most common exception is the viruses because they have either RNA or DNA as a genetic material. So this DNA barcode kya hai is a way of differentiating one organism, one species from another species. So what we are basically trying to see, the basis of variation is the variation in the DNA sequence, right? So we are basically comparing the 16S ribosomal RNA gene. That means the gene which is coding for 16S ribosomal RNA in different bacteria. So if you want to say bacteria A and bacteria B, and let us suppose there is an unknown bacteria. See, in the case of mycoplasma, they do not have cell wall. So as such, there is no gram staining. But still we are saying that they are gram positive in origin. What is the basis? Cell wall ही नहीं है. आप तो gram staining कर ही नहीं सकते हो यहाँ पर. फिर भी आप कह रहे हो कि वो gram positive बैक्टीरिया है. तो बेसिस क्या है? आपने ग्राम पॉजिटिव बैक्टीरिया के 16S राइबोसोमल आरएनए को कोड करने वाले जीन को सीक्वेंस किया और आपने माइकोप्लाज्मा में 16S राइबोसोमल आरएनए को कोड करने वाले जीन को सीक्वेंस किया. जब आपने कंपेयर किया दोनों को तो आपको उनके सीक्वेंस में सिमिलैरिटी नजर आ रही है. एट द लेवल ऑफ डीएनए दे आर क्वाइट सिमिलर, जो हमको उन्हें एक कैटेगरी में कैटेगराइज करने में हेल्प कर रहा है. सो बेसिकली वी आर सीइंग द डिफरेंसेस एट द लेवल ऑफ डीएनए and whether that difference can be used to differentiate one person from one species from another species. Like in a school, different schools have their different uniform just to differentiate one school student from another school student. If you will find any student outside the school campus, by seeing his or her dress, you can say he belonged to or she belonged to this particular school. Similarly, we have the DNA barcode, the sequence of the DNA which we are using as a parameter to recognize, to differentiate one species from the another one. Okay? So this is DNA barcode. Now, in the case of eukaryotic system, we have the most common DNA barcode is the cytochrome C oxidase gene, which is present in the mitochondria. ठीक है, इलेक्ट्रॉन ट्रांसपोर्ट चेन में साइटोक्रोम सी ऑक्सीडेज आपने पढ़ा होगा इलेक्ट्रॉन कैरियर के रूप में. कॉम्प्लेक्स 1, कॉम्प्लेक्स 2, कॉम्प्लेक्स 3, कॉम्प्लेक्स 4. तो साइटोक्रोम सी ऑक्सीडेज 1 जो माइटोकॉन्ड्रियल जीनोम का हिस्सा है, इट इज यूज्ड टू डिफरेंशिएट इन द केस ऑफ यूकैरियोटिक सिस्टम. ठीक है, एज अ डीएनए बारकोड. ठीक है, तो डीएनए बारकोड एक ऐसा सीक्वेंस है जो डाइवर्ज नहीं होगा इवोल्यूशन के साथ, राइट ना. वो चेंज नहीं होगा. किसी स्कूल का ड्रेस अगर आप बार बार चेंज करते रहोगे तो हमें डिफरेंशिएट करना आसान नहीं होगा. एक ऐसी चीज जो उसके जीनोम में कांस्टेंट है विथ द इवोल्यूशन, जो बार बार चेंज नहीं हो रही है. ठीक है, जिसने अपनी कॉन्स्टेंसी को बरकरार रखा है, रिटेन रखा है up to that extent that we could able to differentiate on that basis. All definitely variations are there, but variations are not very large variations and very frequent variations. that is not there in the DNA barcode. It remains that constancy with respect to that particular species. उस स्पीशीज में वो डीएनए बारकोड का जो सीक्वेंस है वो मेंटेन रहेगा इवोल्यूशन के साथ. ठीक है. Okay, so mycoplasma are very specific in their nutrient requirement and they are difficult to grow under laboratory conditions. Due to this particular property, we use a term like fastidious for mycoplasma. Fastidious का मतलब होता है they are very specific in their nutrient requirement. Since we have seen that they are parasitic, so parasitic है मतलब they require a host, right? So उनका जो इवोल्यूशन है वो होस्ट के साथ इवॉल्व हुए हैं. दे मोस्ट ऑफ देम आर होस्ट स्पेसिफिक एंड दे आर इवॉल्व वेरी क्लोजली विद द होस्ट, राइट? सो दैट्स व्हाई देयर रिक्वायरमेंट इज वेरी मच स्पेसिफिक टू द होस्ट इन व्हिच दे आर लिविंग एज अ पैरासाइट. दे आर प्लियोमॉर्फिक और फिलामेंटस. प्लियोमॉर्फिक मींस दे हैव इर्रेगुलर शेप क्योंकि उनके पास सेल वॉल एब्सेंट है. इन सम केसेस दे आर फिलामेंटस आल्सो. Like in the case of spiroplasma, they have a spiral shape, right? And they are difficult to stain because they lack cell wall. They can be killed easily if present outside the host cell through the standard disinfectant. And they are naturally resistant to antibiotics that target cell wall synthesis. तो ऐसे एंटीबायोटिक जो सेल वॉल स्पेसिफिक हैं या उन बैक्टीरिया के लिए है जिनके पास सेल वॉल है, वो एंटीबायोटिक माइकोप्लाज्मा का कुछ भी नहीं बिगाड़ सकते. They cannot do any harm to mycoplasma. Those antibiotics which work specifically at the cell wall or which inhibit cell wall formation, right? Like आपका जो पेनिसिलिन एग्जांपल है, that works on the cell wall. ठीक है, जिसे हम बीटा लैक्टम एंटीबायोटिक्स के ग्रुप में क्लासिफाई करते हैं. ठीक है, लाइसोजाइम है, it also works at the level of cell wall. ठीक है. ओके, फिर एक ये नाम जो माइकोप्लाज्मा लाइक ऑर्गेनिज्म है. अच्छा, जो माइकोप्लाज्मा है उनको आप ल्यूरोनिमोनिया लाइक ऑर्गेनिजम्स भी कहते हो, PPLO. दिस इज आल्सो वन ऑफ द कॉमन नेम ऑफ माइकोप्लाज्मा. ठीक है, PPLO, ल्यूरोनिमोनिया लाइक ऑर्गेनिजम्स. फिर अब हम माइकोप्लाज्मा में ही एक ऐसे ऑर्गेनिज्म के बारे में पढ़ रहे हैं जिन्हें हम कहते हैं फाइटोप्लाज्मा. So phytoplasma को हम क्या कहते हैं? Mycoplasma like organisms. So what are these phytoplasma? Phytoplasmas are the plant parasitic mycoplasma. यानी कि जब आपने host fix कर दिया, मैंने आपको बताया कि mycoplasma can be parasite on human, on animals as well as on plant. So now we are talking of such mycoplasma which have their host as a plant. So phytoplasmas are the plant parasitic mycoplasma and they are the obligate plant parasite. So these are small plant pathogenic bacteria which are limited to the phloem tissues of the plant and they are transmitted by various insects. So what are the characteristic symptoms of phytoplasma related disease? One is phyllody, where the floral structures are converted into leaves. This is also again a retrograde metamorphosis. That means flower एक evolutionary complex structure है जिसमें फीमेल मेल रिप्रोडक्टिव ऑर्गन्स हैं, वो एक सिंपलर स्ट्रक्चर की तरफ मूव कर रहा है, दैट इज लीफ. सो retrograde metamorphosis का एग्जांपल है phyllody. Then witch's bloom, that means clustering of branches. Then reddening of leaves and stem, general yellowing, formation of bunchy fibrous secondary roots. These all are the various symptoms which occur after phytoplasma infection. Okay, then we will go through viruses. So viruses are the connecting link between living and non-living organism. ठीक है, हम उन्हें connecting link क्यों कहते हैं? Does anybody will like to answer why we call viruses as a connecting link between living and non-living organisms?
Because it exhibits both living properties as well as non-living.
बताइए, any living property as well as any non-living property.
When it comes to living, as it multiplies like human cells, in a specific host. When it comes to non-living, if it is not present in a host, like think it is in a environment, it acts like a chemical substance.
Very good.
So we can say that it is
Very nice, very nice, very good answer. So outside the living host, it is simply as a chemical entity. It is just a chemical particle, right? But in the living system, when it enters in the living system, when it enters in the living host, then it will replicate like other living system, right? Okay, so it is a connecting link between living as well as non-living organisms. And people have used Okay, so viruses are very small in size. The range of the size is 20 nanometer to 1400 nanometer. And in order to visualize, in order to determine their size, we need the help of electron microscope, right? They are obligate intracellular parasite. They need a living cell. They live within the living cell, right? And they contain either DNA or RNA as their genetic material, right? Either DNA is a genetic material or RNA is a genetic material. Then the nucleic acid of a virus is surrounded by a protein coat, which is also known as capsid. And the monomeric unit of this protein coat is known as capsomere. So capsomere, they join together, they come together, then they form capsid. Like bricks come together to form a house. Similarly, capsomeres comes together and they form a capsid. Okay, and therefore we also call viruses as nucleoprotein entities. That means they consist of nucleic acid and protein. Genetic material is present either in single stranded state or double stranded state, right? So in the case of viruses, we have the 4 different classes: single stranded DNA, double stranded DNA, single stranded RNA, and double stranded RNA. Can anybody tell me what is the genomic constituent of coronavirus and whether it is single or double?
Single.
It is single.
Single, sir.
It is RNA or DNA?
RNA.
It is RNA, exactly. So coronavirus is an example of single-stranded RNA virus, right? Okay. So genetic material is present either in single-stranded state or double-stranded state. In certain cases, an envelope is also present around these nucleoprotein entities, which usually consist of lipid, protein, and carbohydrate, right? So in certain cases, around the nucleoprotein entity or around the capsid, we have an outer membrane which usually consists of lipid, protein, and carbohydrate. And in most of the cases, this outer membrane is derived from the host cell. Definitely, it is derived from the host cell, but the chemical composition of the envelope is decided by the viral genome. So what this particular membrane contain, definitely it has host component also, but certain protein components which is there in the envelope is decided by the viral genome, not by the host genome. So coronavirus contain envelope. It is an example of envelope virus. It contain envelope also. What is the term virion means? किसने किसी को अनम्यूट करना है? चंद्रकांत जी, आप म्यूट करेंगे? But I am hearing some voice. I don't know. Okay, so a virion is a complete, fully developed infectious viral particle composed of nucleic acid and surrounded by a protein coat, and is a vehicle of transmission from one host cell to another. तो विरियन क्या है? एक कंप्लीट फुली डेवलप्ड इन्फेक्शियस वायरल पार्टिकल है जो न्यूक्लियोप्रोटीन कोट, न्यूक्लिक एसिड और प्रोटीन कोट से बना हुआ है, एंड इट इज अ व्हीकल ऑफ ट्रांसमिशन फ्रॉम वन होस्ट सेल टू अनदर. ठीक है, बट विरियन में एनवेलप भी हो सकता है और वो विदाउट एनवेलप भी हो सकता है, ठीक है। तो ये है एनवेलप एंड नॉन एनवेलप वायरस। तो मैंने आपको कहा कि एनवेलप वायरसेस में वी हैव एन आउटर मेम्ब्रेन एंड दैट आउटर मेम्ब्रेन इज derived फ्रॉम द होस्ट, बट द present in this particular outer membrane, they are decided by the viral genome. So constituent of the envelope में viral genome का भी contribution है. So envelope plus nucleocapsid ये दोनों मिलकर virion बनाते हैं in the case of enveloped viruses, right? Examples में आपके पास envelope virus में क्या है? HIV virus है, HSV virus है, HBV and influenza virus. It is also an example of influenza virus. So they have envelope as well as they have the envelope protein, and this envelope protein is synthesized by using the viral genome. Virus living host cell के अंदर जाने के बाद host cell machinery को utilize कर रहा है अपने component को synthesize करने के लिए, right? In the case of non-enveloped virus, envelope is not there. They have just nucleocapsid. So in the case of non-enveloped virus, nucleocapsid is virion, while in the case of enveloped virus, we have envelope plus nucleocapsid as a virion. इसलिए जब यहाँ पे हम virion की definition पे बात कर रहे थे, तो यहाँ पर envelope की कोई चर्चा हमने नहीं की, but we should keep it in our mind that in the case of enveloped virus, envelope is also a part of virion. Well, in the case of non-enveloped virus, it is just the nucleocapsid. Okay, and viruses have complex structure. There are certain viruses which looks very simple to you, while there are certain viruses which have a complex structure. For example, if you see here, in this case, bacteriophage, see, they have a complex structure. They have a head, then they have a tail, then they have fibers here. This is the neck region. So they have some complex structure here. But in the case of influenza virus, if you will see, they are not so complex like that. Simple nucleocapsid is there, then outer membrane is also there, protein से बना हुआ coat है, capsid है, और अंदर DNA present है. So it doesn't look so complex as the bacteriophage. Okay, then we have to go through fungi also. So fungus, it's definitely a eukaryotic cell, and like other eukaryotic cell, fungus में true nucleus present होता है, well membrane or membrane bound organelles are also there, cell wall is also present. Cell wall in the case of fungi is made up of chitin, right? Then fungal cells को हम divide करते हैं morphologically into two types, multicellular filamentous fungi and unicellular fungi. So multicellular filamentous fungi, they consist of true hyphae, and hyphae are elongated thread-like structure which may be septate or aseptate, right? So consist of true hyphae. Filamentous fungi से हमारा मतलब है कि they have hyphae, and hyphae are the elongated thread-like structure जिनमें septa present हो भी सकता है और septa present नहीं भी हो सकता है. Well, in the case of unicellular fungi, they have pseudo hyphae. They do not have true hyphae, right? So pseudo hyphae से हमारा क्या मतलब है? That means if you have seen the example of budding yeast, let us see this diagram. Here, this is the budding yeast cell. So the cell has not divided completely, right? It has joined still with the parent cell. So this particular structure, this budding yeast, then from this particular cell you may have another, then from that you may have another, but they are not truly septate. Definitely we are seeing small fragments on the parent cell, but they are not true hyphae. So this budding yeast cell is an example of pseudo hyphae. This you can see here, budding yeast with pseudo hyphae, right? So a single yeast cell से आपको इतना सारा ब्रांचिंग होता जा रहा है, इतने सारे नए स्ट्रक्चर बनते जा रहे हैं, बट ये नया स्ट्रक्चर ट्रू हाइफे को रिप्रेजेंट नहीं करता है, ठीक है। वेल इन द केस, हाँ, देन हाइफे के सिस्टम में अगर हम बात करें तो वी हैव बोथ सेप्टेट हाइफे एज वेल एज aseptate hyphae. So in the case of aseptate hyphae, we do not have any partition, while in the case of septate hyphae, we have the partitions. And these hyphae together, they will form mycelium. So this is the basic structure of a fungi. So I think so this is my last slide. Yeah. So if you have any doubt, please let me know. अगर किसी को कोई डाउट हो तो आप पूछिए मुझसे. फिर मैं आपको और भी कुछ बताता हूं. यहां तक आपको अगर कोई डाउट हो तो आप पूछ सकते हैं. इफ यू हैव एनी डाउट प्लीज आस्क.
Hello, sir. How can we differentiate between pseudo hyphae and septate hyphae, sir?
Pseudo hyphae and septate hyphae. Okay. In pseudo hyphae, the constrictions which are there, you can see here, we do not have a septa. Actually, it is we cannot able to see in the B diagram, but they do not have a true partition between the cells. They are still connected with each other. जैसे अगर आपने कभी बलून देखा होगा और बलून में अगर आप पानी भर लेते हैं और पानी भरने के बाद आप बलून को कंस्ट्रिक्ट करते हैं, इवन हवा भरने के बाद भी आप कंस्ट्रिक्ट करते हो, तो वो जो कंस्ट्रिक्शन आपने किया उसको बलून को फोल्ड करके या उसको ट्विस्ट करके, इट इज लाइक अ बडिंग, इट इज लाइक अ स्यूडो हाइफे. ठीक है. जस्ट यू हैव प्रेस्ड फ्रॉम वन साइड. नाउ आपको बाहर से ऐसा दिखाई दे रहा है दे आर टू बलून्स, बट वेदर दे आर ट्रू टू बलून्स? No, it is a single balloon constricted. So ये जो आपको pseudo hyphae है, यहाँ पर आपको ये constriction है, but there is no wall formation, there is no septa, there is no septate structure is there. ठीक है. Partition as such physical partition नहीं है, सिर्फ constriction present है, जिसकी वजह से आपको लग रहा है कि एक और नई चीज बन गई है, but actually नया बना नहीं है. वो original सिर्फ constrict होकर आपको दो दिखाई दे रहे हैं. कुछ समझ में आया? क्लियर हुआ?
हाँ सर, हाँ सर.
ओके, ठीक है.
सर सी चार्ट बॉक्स सर.
ओके फाइन. चैट बॉक्स, जस्ट अ मिनट. ओके फाइन. Do autotrophic bacteria also act as heterotroph in any stage of their life? For example, in E. coli. But E. coli is not an autotrophic bacteria. Do autotrophic bacteria? I don't think so. I don't think so. An autotrophic bacteria also act as a heterotroph. I am not sure about it, but I think so. It's not there. If a bacteria is autotrophic, I think so. It is autotrophic, but let me confirm. Otherwise, I will convey it to Bhale Rao sir if I will get an answer. Okay. Any other question? So you all are preparing for the JRF exam.
हाँ सर, नेट
नेट
ARS
ARS
JRF
also.
SRF.
Okay. So
हाँ,
आप बताएंगे आप कैसे तैयारी कर रहे हो, so that I can help you. क्या क्या बुक्स पढ़ रहे हो
हाँ
आप?
सर, just we started sir, we are in MSc first year.
आप अभी MSc फर्स्ट ईयर में है and your specialization
हाँ,
is biotechnology. Your
Biochemistry sir.
specialization is biochemistry. Okay. So you have to.
हाँ sir, but
बताइए.
sir, but in this group, crop physiology, biotechnology,
Okay. Everyone is there.
genetics, all are
there.
present. हाँ sir,
Okay.
regarding
Regarding plant sciences. So आप which books you people usually read and like what is your strategy? First, you tell me how you have thought of starting since you all are in MSc first year. तो अभी क्या सोचा है, कैसे क्या स्टार्ट करना है, क्या जानकारी है अभी आपके पास, थोड़ा सा मुझे बताओ.
plant sciences. Sir, as we are in first semester, this training program started, sir, from February 15th. So we are getting experience from teachers like Leninzer, Strayer book to refer for biochemistry.
Okay.
Sir. So we are trying to start.
Okay, that's very good. So what I will tell you that you first take out the syllabus of ARS exam, whichever stream from you are, and try to read every topic thoroughly. If your purpose is just ARS net, then you need not to be so thorough. But first of all, take out the syllabus, write and go through all the topics one by one, complete each topic. Try to have good information of all the topics. Now, from where you will get information? I am from biotechnology, so I will tell about biotechnology. Even I can tell about biochemistry also. We have very good English authors book you may have in your library also. So try to understand the concept first. आप कॉन्सेप्ट को अगर अच्छे से समझ लेंगे तो आप ऑब्जेक्टिव क्वेश्चंस भी लिख पाएंगे और सब्जेक्टिव क्वेश्चंस भी लिख पाएंगे. You can able to attempt both subjective as well as objective questions equally well if your concept is good. So in order to make your concept good, you have to start reading from good books. Good books means the books which have well illustrated diagrams. Through those books, you can visualize the things very clearly. See, in life science, even in genetics also, we have to visualize the things. We cannot see DNA always. We cannot see biomolecules always through our naked eye. So we have to visualize what is happening inside the cell at the biochemical level, what is happening at the DNA level. So in order to visualize all these biochemical and molecular process, you must have to go through those books which have very nice illustration of diagrams. When you start reading those books, maybe at first you feel certain difficulty. Maybe the English they have used may not be so much acquainted, but when you start reading it slowly, again and again, then you will be able to clear your concept. So you start reading topic wise. And now information is not at all dearth. Lot of information is there. Even in internet also, you can read through YouTube also. You can go through different books also. Through internet, various e-books are there. आप सारा मटेरियल अपने पास रख लीजिए and start कीजिए one by one. And one important thing, after having all these lectures, अगर आपके stream का there is another colleague, there is another batchmate from the same stream, start discussing with each other. Most of the times we feel that if we share, we will lose. But it is actually just opposite. If we will share, we will gain. We will come to know whether our concept is really at the right in the right direction, or still we need certain more information to make our concept further strong. When you start explaining someone, then you come to know whether you are thorough with that topic or you are not thorough with that topic. So try to start discussing among yourself. जब भी आपको टाइम मिलता है वीक में एक दिन, स्टार्ट करो डिस्कशन करना, ठीक है? इट इज वेरी 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 मच इंपॉर्टेंट. हम ज्यादातर ये सोचते हैं कि हम अपने नोट्स, अपनी नॉलेज को बचा कर रखेंगे, हम शेयर नहीं करेंगे, बट हम जब शेयर करते हैं तो हम ज्यादा अच्छे से चीजों को समझ पाते हैं. तो एक तो वो इम्पोर्टेंट है. फिर अगर आपका पर्पस ARS नेट का है, आपको ऑब्जेक्टिव एग्जाम्स, ऑब्जेक्टिव है, मल्टीपल चॉइस क्वेश्चंस हैं, तो उसके लिए कुछ अच्छे जो मल्टीपल चॉइस क्वेश्चंस आपको मिलते हैं. जैसे जैसे आप टॉपिक रीड करते जाते हो, लेट अस सपोज टुडे यू हैव रेड अबाउट DNA क्लोनिंग, ठीक है? देन इफ यू विल सर्च ऑन गूगल मल्टीपल चॉइस क्वेश्चंस ऑन DNA क्लोनिंग, you will get number of multiple choice questions. So whatever you have read, try to check whether you have absorbed the same from the book or still you are lacking, so that you will come to know after going through the questions. If you will be able to understand the question well, if you can able to understand the given options well, and if you are doing it right, that means you have understood the concept well, you have visualized it well. But अगर क्वेश्चन अटेम्प्ट करने पे क्वेश्चन समझ में नहीं आ रहा है, this question, I have read entire book of Leninger biochemistry, but still where this particular question have come from, that means इसका मतलब है maybe the book but I haven't understood the concept, maybe something is still missing. So try to go through questions which are available on the internet. Try to have the good questions, like good sites for the questions, and you will come to know most of them are okay, most of them are fine. अगर गलत होगा तो भी आपको पता चल जाएगा, but try to go through questions. जितने बार जितने ज्यादा क्वेश्चन सॉल्व करेंगे उतना अच्छा विजुअलाइजेशन होगा. इवन लेनिंजर की बायोकेमिस्ट्री की बुक में भी ऑन द बैक साइड ऑफ ईच चैप्टर यू हैव क्वेश्चंस. एटलीस्ट ट्राई करिए, गो थ्रू इट. इवन आप CSIR JRF के लिए भी अगर तैयारी करते हैं फॉर PhD, तो आपके लिए CSIR JRF आप क्वालिफाई कर लेंगे, नाउ द फेलोशिप्स हैव इंक्रीज्ड, तो आपको बहुत हेल्प हो जाएगा. तो एक तो हमको सबसे पहले जिस भी एग्जाम की हम तैयारी कर रहे हैं, विच एवर एग्जाम, फर्स्ट ऑफ ऑल यू नीड टू नो द सिलेबस, राइट? वंस यू नो द सिलेबस, then you have to go through each topic of the syllabus from the good books, in which you will get more number of diagrams, better visualization. ठीक है? And third, after reading each chapter, after reading each concept, try to solve questions from the internet or from any other book if you have in your hand. Try to solve the questions from each concept node that you have understood. चाहे वो restriction enzymes हो, maybe it is southern hybridization, maybe it is northern hybridization, maybe it is transcription, translation. Try to go through the questions. तो उससे आपको clarity और ज़्यादा हो जाएगी. And whenever you are reading anything, try to visualize what you are reading. If somebody is saying that RNA polymerase sits on the promoter and then start transcription, so don't just read it randomly or vaguely. Just try to visualize in a living cell. We are talking about the nucleus, because in the nucleus we have a DNA, in the nucleus we have a RNA. Now RNA polymerase is going and it is binding to the promoter sequence. Then it will start transcription from the transcription start site. So if you start visualizing what you are reading through your daily examples, even of your life, very common example, है ना? Then it will be far better for you. So start visualizing the things, and that you can do while you are reading. If somebody says we have a restriction enzymes of two types, we have blunt end ligation, we have sticky end ligation. Then what is sticky end ligation? Try to visualize. We have the overhangs there. What overhang look like? It looks like this. This is, they are not on the same line. This is another overhang. If we will do sticky end ligation, it will be like this. If we will do blunt end ligation, it will be like this. So try to start visualizing the things, then it will be far easy for you to remember. Even polymerase chain reaction. What is PCR? What is happening in the PCR? What is primer? Try to visualize primer. It is a short DNA fragment. What it is actually doing? जब आप क्वेश्चन पूछने लगोगे अपने आप से, when you start questioning, then you will gain a lot of knowledge. बाकी JRF एग्जाम क्लियर करने के लिए या ARS साइंटिस्ट बनने के लिए, कुछ भी करने के लिए ऐसे कुछ बहुत अलग से करने की जरूरत नहीं है. बस जैसा आप पढ़ाई कर रहे हो उसके अप्रोच को थोड़ा सा आप अगर इम्प्रूव कर लेते हो तो there will be no problem. If I have certain memory based questions of ARS net exam of biotechnology, I will mail it to you, sir, so you can have the printout and you can go through them, but they are memory based and now it is entirely different. So the level of exam is quite tough nowadays and they ask more of practical orientation also. So practical orientation आपको तभी आएगा जब आप चीजों को विजुलाइज करने की कोशिश करोगे. Let us suppose your MSc seniors are there, they may be doing work in the lab, you are from MSc first year, you can go along with them. Maybe they are doing DNA isolation, at least you can follow what they are doing. Sir, okay, we are using liquid nitrogen for the purpose of grinding. Then you have one question, why liquid nitrogen? Then we have used CTAP. Why CTAP? We have used isopropyl alcohol for the purpose of precipitating DNA. Why is it so? Some people are using 100% ethanol. At least you will be get acquainted with the terms, with the name of different chemicals that they are using. So try to go along with them, what they are doing. Be curious what they are doing, what research topic on which they are working, किसके ऊपर काम कर रहे हैं. एक बार पूछिए, उनका टॉपिक नोट कर लीजिए. घर आने के बाद अपने इंटरनेट पर सर्च कर लीजिए, भाई what are the different words in that particular topic. So this is the way you can increase your knowledge and then it will be very easy for you to qualify any exam. So if you have anything to tell me. बताइए.
Sir, see chat box, sir.
Sorry. Okay, I will share my contact number. Shall I write here? ठीक है. आप लोग CSI JRF की तैयारी भी कीजिए. इवन दिस ईयर दे हैव 2 more exams to go. एक एग्जाम तो कंप्लीट हो गया है, 2 एग्जाम अभी और होने बाकी हैं. ड्यू टू कोविड पेंडेमिक दे हैव डिसाइडेड टू कंडक्ट 3 exams in a year. So if you have a chance to give that exam, try to give. सब कुछ सिलेबस में पढ़ना CSI के केस में डिफिकल्ट है, बट जितना आपको अपने से अच्छे से आता है ना, अगर आप उतने पोर्शंस को भी कवर कर, इवन आप CSI आराम से क्वालिफाई कर पाओगे. ARS के एग्जाम का सिलेबस उतना ज्यादा नहीं है और ARS is subjective as well as objective. So in that you have to read, you have to go through completely. But in the case of CSI, you have so many life sciences discipline included. So in that, no need to go through every, like ecology, immunology, then environmental biotechnology, then marine biotechnology, then evolutionary biotechnology. So many things are there in that. बट फिर भी एक मेजर प्रोपोर्शन जो आप पढ़ते हो अपने एग्रीकल्चर में, या मान लीजिए आपका बैकग्राउंड BSc बायोटेक्नोलॉजी भी है, तो उस चीज को अच्छे से रीड करके जाएंगे तो आप आराम से एग्जाम क्वालिफाई कर सकते हो. बट प्रैक्टिस करना स्टार्ट करो और आपस में डिस्कशन करना शुरू करो एक दूसरे से इन अ वेरी फ्रेंडली मोड. ठीक है. डू नॉट hesitate in a very friendly mode without any ego and anything. It's a learning phase for all of you. Even for me, it's a learning phase. If I will keep a ego with me, I will not able to learn anything. तो आप लोग भी किसको क्या आता है, किसको क्या पता है, उससे हमें कोई लेना देना नहीं है. हमें जो आता है वो हम बताने की कोशिश कर रहे हैं, उसे जो आता होगा वो बताएगा, जैसे कि हम दोनों की ग्रोथ हो सके. कोई किसी एग्जाम में निकल जाएगा, कोई किसी एग्जाम में निकल जाएगा, दोनों भी एक एग्जाम में निकल सकते हैं, है ना. तो इंटेंशन अच्छा रखकर डिस्कस करना शुरू करो. और कोई डाउट हो तो टेल मी इफ यू हैव एनी डाउट. Actually, this was not from my subject, this particular topic, but सारा टॉपिक कवर हो गया था, तो फिर मुझे ये टॉपिक में, so I have prepared slide. Maybe they are not very comprehensive, but I will improve them further and I will give it to sir. I will mail it to him. तो आपके पास पहुंच जाएगा ये पावर पॉइंट प्रेजेंटेशन भी, थोड़ा और बेहतर करके मैं इसे दे दूंगा उनको. और कोई पूछेगा कुछ? Any doubt? How is library there? Acha, one more thing I have to tell. Okay, this is a PDF drive. Anybody know about this? कोई जानता है इसके बारे में? Anybody of you know about this PDF drive? Chinta?
No, sir.
Okay. So, actually, this is, if you will type in Google PDF drive, it will open and you can able to browse many books, and so many molecular biology books you will able to get through it in online. Like you can download and you can keep it in your laptop or in your PC. And if you need to take a printout, you can take a printout also. Or if you go like that, you can read like that. You will get most of the molecular biology groups books with latest editions. If not latest, at least just previous edition. चाहे लेनिंजर का बुक हो, चाहे तेज एंड जीगर हो, चाहे बुकनिन हो, चाहे वॉइट एंड वॉइट हो, एनी बुक एंड फ्रॉम एनी डिसिप्लिन, नॉट जस्ट मॉलिक्यूलर बायोलॉजी, एनी डिसिप्लिन का बुक अगर वो वहां पीडीएफ ड्राइव में अवेलेबल है तो आप डाउनलोड करके भी उससे पढ़ सकते हो. और कोशिश करो जितना ज्यादा आप बुक्स को पढ़ सको. नॉट बिकम कंटेंट जस्ट बाय रीडिंग थ्रू वन बुक. कि आपने बीडी सिंह पढ़ लिया, बीडी सिंह बायोटेक्नोलॉजी कंप्लीट कर लिया, बस ऐसा नहीं है. बीडी सिंह बायोटेक्नोलॉजी अगर पढ़ लिया है, इट इज अ बेसिक बुक, आपको एक बेसिक जानकारी हो गई, देन ट्राई टू मेक योर लर्निंग बिट एडवांस्ड. अभी क्या है ना, नाउ द एग्जाम्स दे हैव गिवन इट टू डिफरेंट एजेंसीज व्हिच आर कंडक्टिंग एग्जाम. देयर एग्जाम का जो लेवल है वो थोड़ा इम्प्रूव हुआ है पहले से. तो ऐसे बुक्स को पढ़ना शुरू करो, ज्यादा से ज्यादा बुक्स को पढ़ना शुरू करो. जिस कोई प्रैक्टिकल मैनुअल का बुक है उसको भी देखो क्या चीज है, तो आपको ज्यादा अच्छे से सॉल्व कर पाओगे क्वेश्चंस को, ज्यादा अच्छे से चीजों को समझ पाओगे. 3 इडियट्स का मूवी देखा है ना सबने? जहां से भी ज्ञान मिल सकता है वहां से ज्ञान लेने की कोशिश करना है, बस यही है take home message. So from wherever you can get the knowledge, try to get it and try to write it and revise it. एक बार बुक पढ़ने के बाद फिर बुक को बंद करके रख दिया, so it will not help you. You have to keep reading, keep writing, keep revising. And how you will revise? When you will discuss with someone. This is a sort of revision only. आपको रिविजन हो जाता है कि मैंने जो पढ़ा है वो ठीक से पढ़ा है या नहीं पढ़ा है. ठीक है? So this PDF drive you can use. This is a very good site for the book. Just in the Google you write PDF drive. आपको बहुत सारी बुक्स मिल जाएंगी. So और कुछ? Anything else from my side? बताइए। इतना इंफॉर्मेटिव तो नहीं है ये, मुझे पता है, बिकॉज आई एम नॉट वेरी वेल वर्स विद दिस टॉपिक्स अभी राइट नाउ, बट फिर भी मैंने थोड़ा कोशिश तो किया कि आप लोगों को कुछ तो समझाऊंगा, बट आई विल इम्प्रूव दिस स्लाइड्स एंड आई विल मेल इट टू योरसेल्फ। So,
No, sir, it is well explained with diagrams, sir. It is very nice, sir.
चलो ठीक है, तब तो अच्छी बात है, बट मैं इसे और अच्छा करके आपको मैं मेल कर दूंगा, और मैं पेपर्स भी आपको मेल कर दूंगा या सर को मेल कर दूंगा, जो भी मेरे पास पुराने
हाँ
पेपर्स हैं।
सर,
इट्स मेमोरी बेस्ड, इट इज नथिंग लाइक द ओरिजिनल पेपर, किसी ने बताया क्या क्या आया तो हमने लिखा हुआ है, सो आई विल मेल इट टू सर।
हाँ, okay, sir.
Okay,
Okay.
so what is the time for lunch for you all there? At 1 o'clock?
हाँ, 1 to 2,
1 to
sir.
2, ठीक है, so भालेराव सर कहाँ पर?
हाँ, shall
we close?
हाँ,
Sir,
हाँ, sir. But Balarav sir assigned with some other work,
अच्छा,
sir.
नो प्रॉब्लम, कोई दिक्कत नहीं
हाँ,
है,
हाँ,
you
sir.
all take care and my number is there with you
Yes.
and I will give my
हाँ,
mail
sir.
ID also, so if you have, these all are in small. 1 minute. ठीक है, तो आपको कभी भी कुछ भी है तो आप मुझे मेल भी कर सकते हो और कॉल भी कर सकते हो। जो 26 वाला नंबर है दैट इज व्हाट्सएप नंबर, व्हाट्सएप करके भी कुछ पूछ सकते हो, ठीक है। So
हाँ, sir.
all
Thank
the
you, sir.
best to all of you.
Thank you,
Keep
sir.
reading and keep discussing with each other.
हाँ, sir.
Okay.
Thank you from participants
Okay,
side,
okay.
sir.
Thank you so much. Thank you so
हाँ,
much.
sir.
Okay.
Thank
Okay.
you, sir.